me show you how to be a good breezy and change your predictions after.
Introduce you. Hi everyone, welcome to this third Learn Based Stats webinar. Today we have the chance and honor to welcome back Justin Bois to the show. I know a lot of you will be happy because a lot of you really loved Justin's episode on Learn Based Stats. So today we have a very interesting and detailed webinar about biology in the Bayesian world. And without further ado, I will give you the floor, Justin. Maybe quick introduction about you, what you do, and then you can start with the presentation, because if people are interested in your background and your whole story, well, there is the whole podcast episode for that, and I will put a link right now in the chat.
Thanks, Alex. So just then when you were introducing me, you froze for just a second, which also made my heart freeze. So hopefully there won't be any problem with that. If there is, I'll just because I just saw that in case I freeze, I should be right back. Okay, so yeah, my name is Justin Bois. I'm a teaching professor in the Division of Biology and Biological Engineering at the California Institute of Technology. That's in Pasadena, California. It's commonly known as Caltech. I'm sitting in my office there at Caltech right now. I chose to sit in my office instead of at my home because of the extra fast and reliable internet connection here. Let's hope that holds up. One of the things that I wasn't quite planning on is that if I look out my window right now, I see a truck that says Facilities Management. So that means we may occasionally hear a bang or something from outside. They seem to be doing a little bit of heavy work out there at the moment, but I don't think it'll be too distracting. Okay, so my job here at Caltech is as a teaching professor. I'm actually the only teaching professor in our entire division of biology and biological engineering. All of our other faculty, well, I should say I'm the only full-time teaching faculty. All of our other faculty primarily run research groups. And I do engage in some research, but it's almost always through the students that I have through my classes. And it's often involves doing some data analysis of some of the data sets that they are acquiring in their laboratory work. And so it really provides me the opportunity to do often a Bayesian approach to lots of different data sets across the biological sciences, which is kind of a gift. I get a chance to really apply these techniques that I'm guessing because you're here, that we all know and love to a wide variety of topics. And so what I'm going to talk about today is I'm really going to do a case study, and I'll talk about what the case study is in just a moment. But I just want to talk about sort of what the main points I want you to take away from this webinar are. So first, as I go through this, I'm going to emphasize the importance of domain knowledge. What I mean by that is that when you're analyzing a data set or constructing models or doing any sort of modeling, it's really important that you are well grounded in really all of the background of the system that you're studying. So let's say that I want to study, say, some sort of pathology, say a disease, and I have some data about that. Well, I need to be discussing with pathologists, with immunologists. I need to make sure that I actually fully understand the system so that I can, really properly model it. And there's really, and I guess for practicing data scientist type of role, where you're going to be doing that type of work, where you're basically providing data science support for a broader goals of a company or a group within your company or something like that. It's really important that you connect with the people who really understand what it is that you're trying to do, that you don't just say, give me your data, then I'll do an analysis and I'll give it back to you. The only time you can do that is if you deeply already understand what it is what the system is that you're trying to understand. I'm also going to talk a little bit about some approaches to coming up with priors. This is a famous topic to talk about in Bayesian analysis, and I'll talk about some of the things that I like to do to really try to mathematize what it is that I know about parameter values going into an analysis. That's ultimately what a prior is. It's a mathematization of what we know going in. And so I'm going to talk a little bit about how we can do that. And I think I might even put Alex on the spot just for fun when we do that. I also go over a few different tips and tricks for sort of keeping your analysis organized. And then finally, as we go through it, you'll see a demonstration of some of the tools that I like to use as I go through a Bayesian workflow. So basically, the way I've organized this is we're just going to do a case study, and as we go through, these topics are going to be going to keep popping up, but I wanted to kind of lay them out for you before we get going, so that you kind of see them when they come. Okay, so let's jump into the case study, and what I'm going to do is I'm going to provide you with a little bit of domain knowledge going into it about what it is that we're studying, and we're going to be studying a biological structure, a cell biological structure called microtubules. Now, microtubules are essential components of the cytoskeleton. The cytoskeleton is the sort of these filaments that are like struts that give a cell its structure. And if you look at this image that I'm showing here, you'll see labeled in, there are three different colors. There's red, blue, and green. Now, the blue is what's called DAPI, and what that does is that stains DNA. So the blue is going to show you where the nucleus is, because that's where the DNA lives. And so we're looking at it. Looks like 1, 2, 3, 4, 5 cells. This one in the middle is a really big cell, and so the blue is the nucleus. Now, the red is one of the cytoskeletal proteins, one of the dominant cytoskeletal proteins called actin. And you can see there's like these red filaments there that are also giving some of the shape. And you'll see that the red is really enhanced on the periphery of the cell. And in fact, that's what's called the cell cortex. And this gives the shape to the outside part of the cell. Now, in green, those are the microtubules. And those are the things that we're going to be working with today. And you can see that the microtubules also have this filament-like structure. You can see that they're highly organized. You can see that they're sort of bundled into thicker struts as it goes across. If you go, say, top to bottom in that cell in the center. And they're really important structures for organizing a cell. Now, importantly, these things are very dynamic. And in fact, it's the dynamics of these things that we're going to be studying. And so just partly because it's utterly beautiful and one of the most beautiful things I've seen in nature, but also to give you a view of how dynamic these microtubules are, I'm going to show you this movie. So this is the one cell embryo of a nematode called Caenorhabditis elegans. So the total distance across this image is about 50 microns. That's 50 micrometers. And this is, so it's an egg, and it's got one cell, and you'll see these two pink circles. Those are the two nuclei. One of them was donated from the egg, and the other was donated from the sperm. And in white, what we're going to see throughout this thing, that is tubulin, which is the components that make microtubules. And so the microtubules are going to be in white in this image. And what you're going to see is the first cell division of this embryo. And you're going to see that the microtubules are really the prime movers in this process. So just take a look and enjoy the beauty. So we have this migration of these two nuclei coming together. You can see the DNA is condensing inside them and to form chromosomes. The microtubules form these structures, and as they push against the outside of the cell, they center these now fused nuclei. The microtubules attach to the DNA in the middle, the chromosomes in the middle, and then are involved in pulling it apart. And then you see that dramatic oscillation where they rock back and forth prior to the cell division. Then you can see they actually start to form for a second cell division coming up pretty soon. And all of that is driven by microtubules. And so to study the structure and dynamics of these things is really to understand how fundamental processes in cells proceed. So if you look at a cartoon of what the tip of a microtubule looks like, so they're constructed from these little smaller units called tubulin. And you can sort of, as you look at that image, you see that the tubulins look roughly like rectangles. At the top, the top part of the rectangle is red, the bottom part's green. And then the next one down is blue and green. And these things polymerize, so they grow on the end, and they form these tube structure. That's why they're called microtubules. But what happens is sometimes, instead of growing, they actually suddenly split apart like that, and they depolymerize, or they get destroyed, they shrink. That is called, there's a name for that, it's called microtubule catastrophe. So watch this one more time. So they're growing, growing, growing. They keep adding things to the end, so they're getting longer and longer and longer. But eventually, something gets triggered, and it rapidly depolymerizes and splits apart, and that's called microtubule catastrophe. That is the process that we're going to try to understand today, which is the process of microtubule catastrophe. And we're going to analyze data that comes from a very clever experiment. This experiment was done by Melissa Gardner, who's now a professor at the University of Minnesota, and Maria Zonic, who's now a professor at Vanderbilt University. They did this in collaboration with each other when they were both postdoctoral scientists at the Max Planck Institute of Molecular Cell Biology and Genetics in Dresden, which I'm actually going to visit in about 3 weeks to teach a statistics course. This is one of my favorite experiments. So what they did is
so to make sure I understand, the catastrophe of the microtubules, why are we interested in that right now? It's because we know it happens, but we don't know why?
We know it happens. We're still working on understanding why. But it is important that it does happen because that's part of how, for example, the size of the mitotic spindle, the thing that's going to pull apart the chromosomes, it's how it's an important part of how that size is regulated. Similarly, by undergoing catastrophe, a microtubule can actually exert a pulling force on something that can be attached to the end, and it can slide with it as it depolymerizes. So catastrophe is actually very important in many cellular processes.
So so it's not that negative, because catastrophe sounds like you would not want that, but actually you could want that actually in a lot of contexts.
Yes, and in fact, maybe some of you have heard of these injuries that people who play American football or hockey have, where they get plaques inside their brains. And maybe you've heard of a protein called tau, which is involved in forming these plaques, and that's what leads to some of the dementia and the problems that a lot of these athletes have from repeated head injuries. It turns out that the microtubules are very, very important in structuring cells in the nervous system and maintaining synapses and that sort of things. And synapses themselves are also dynamic. And what happens is that if they get stabilized, in other words, these plaques of tau protein prevent them from undergoing catastrophe, from depolymerizing, that is actually what leads to the pathology of that of those diseases from the head injuries. So catastrophe is actually very important. It needs to happen. But it's one of those, there's all sorts of quirky names in the biological sciences, and catastrophe is one of them. So the experiment that Melissa and Maria set up is they actually took advantage of sort of what I just described. These tau proteins, they can stabilize part of the microtubule by having these things bind to it so it doesn't depolymerize, and that's shown here in red. And what they can do is they can then attach that through biochemical means to the glass, like a glass slide that they can then put on a microscope. That's what those little Y-like things at the bottom of the red part are. That's a biochemical attachment to the glass. And then what they can do is they can put in these little tubulin things that we saw in the cartoon that polymerize it, and they can watch individual microtubules polymerize by doing a microscopy technique known as total internal reflection microscopy, the acronym being TIRF. The F is for fluorescence, total internal reflection fluorescence microscopy. And that's how they can watch these microtubules grow. Now, if we look at, this is a still image from their experiment. Actually, it's a related experiment. Where you can see in red, that's the stabilized part, and then you can see the green part growing or possibly shrinking. So you can watch how these green parts evolve over time. Now, I'm going to show you a movie, and I want you to focus on, so you see these so again, the images are kind of noisy. It's a very challenging microscopic technique. But if you look at the top microtubule, it's the one, so it's between the timestamp, which is one minute there, and then the concentration of tubulin. It's between that, and just go down, you see it's sort of at a slant, like at this angle. Now we'll watch this, and you'll see that it's going to grow, and then boom, depolymerize. You see that? So it grows, 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 and then boom, depolymerize. This movie's on a loop. Grow, 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 depolymerize. Grow, 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 depolymerize. So that switching from growth to depolymerization, that is catastrophe. So this is a movie of catastrophe. Now, the way in which you analyze this, and Maria and Melissa spent forever doing this, is that they did hand segmentation. So they did this before all these AI stuff. They actually would draw a line of pixels along a microtubule, and so what you're looking at here is each row of pixels in this image is a row of pixels along a microtubule, and then as you go down, they're stacking different time points. And so what you're seeing, the red is the stabilized part of it. So that's not going to depolymerize. And then the green triangle looking thing, that's that microtubule growing over time. So remember, time is going down the axis. And then the green is the microtubule growing, and you see it grows. And then you see that, boom, catastrophe happens, and it very rapidly shrinks back to the stabilized seed. And they can do this over and over again, and they can look at different catastrophe events. So if you look at event 1, that grew for about 250 seconds. Event 2 grew for maybe 300. But event 3, it only grew for, maybe 50, 60 seconds, something like that, right? And so they basically did this experiment over and over again, where they measured how long does it take from starting to grow off the seed to undergo catastrophe. Because remember, when the microtubule is not undergoing catastrophe, it's growing. Those are the only two things it can do. It doesn't ever just sit there. You can see that from these plots here. And so that's what the data set is. It's basically a list of times for how long did it take to undergo catastrophe. So that's what the data set is. It's a difficult experiment, painstaking experiment. What they got was basically it's a list of times or how long it took between when it started to grow to then undergo catastrophe. Because I'm assuming, because Alex is nodding, that you're all nodding too, so that's clear what the experiment is. And so that will help us now go into the modeling.
Mm-hmm.
All right.
Yeah, and of course, any question on that, folks?
Let's make sure we understand the experiment before we move on. Yeah.
Any question on that? Let us know in the chat. Otherwise, we're going to assume that it was clear and understood.
Oh, great. Thank you.
Okay.
So now I know what's going to happen is that you're all going to be like, this Bayesian stuff's really cool. Now you're all going to say, you know what? I want to be a cell biologist because this is so cool. But the cool thing about that is that when you become a cell biologist, you can use your Bayesian skills. It works out perfectly.
Yes.
Okay.
Yeah, perfect. So thanks, Justine, and thanks, Janine and Matt, for letting us know.
Yeah, thank you. Okay. So this is the experiment, but we have to, the next question is, all right, well, what are we trying to learn from this experiment? Mm-hmm. Yes, we've watched these catastrophes happen over and over again, but what is it that we're trying to learn? Why would they do this experiment in the first place? Mm-hmm. And there's something to be said for exploratory research, just looking at things, right? And that's great. But let's remind ourselves what Darwin said. Darwin said that any observation should be for or against some view if it is to be of any service. And I think this is a little stark. And so what I'm going to do is I'm going to show you sort of an updated quote of this from my colleague, Rob Phillips, that sort of explains this a little bit more carefully. But I think really what Darwin is saying here is that implicit in any observation really should be some sort of a model. And here's how Rob Phillips puts it. He says, quantitative data demand quantitative models. I think that's a really important idea. Quantitative data demand quantitative models. If we're scientists and we're making measurements of things, we're trying to learn something that's pretty general. And our framework for thinking is within models. And conversely, quantitative models need to provide experimentally testable quantitative predictions about biological phenomena. In other words, if we're going to be making some measurements, we really should have a generative model in mind. And that experiment should provide us parameters for that model, or that experiment should provide tests of that model to see if that model actually is commensurate with what we observe in the system that we're trying to learn about. And so that's really why I think that a lot of these types of measurements that I encounter in the biological sciences are really amenable to Bayesian modeling. because the model, especially in a Bayesian context, and I should be clear, parametric Bayesian modeling, because in a Bayesian framework, the model assumptions are very, very clear. They have to be stated explicitly, and we can really identify what is the parameters that we're trying to measure. So let's think back to this experiment where we have these times to catastrophe. And we might want to think about, okay, so let's think of a generative model for this. Now, we don't actually know all of the details of the biochemistry that's happening at the tip of this microtubule. Yes, there is some knowledge. I won't get into it too much. It has to do with ATPs and ADPs and phosphates and all this stuff. We won't get into what is known about the chemistry, except to say that the waters are murky. And so one simple question that we can ask on this is, well, okay, if I'm going to grow a microtubule, something's going to trigger a catastrophe. And one important piece of domain knowledge that I think is really essential for modeling almost anything that's related to a biochemical process is that across the whole world of chemistry, chemical events are very, very effectively modeled as Poisson processes. As a reminder, a Poisson process is a memoryless process, meaning that when the next event happens is totally independent of when the previous one occurred. And that makes sense in a chemical sense because what's happening is biomolecules are zooming all around by diffusion, and then it's some sort of random collision or some sort of random conformational change due to rotational diffusion or vibrational modes or whatever that's going to lead to a biochemical change. And then that is well modeled as a Poisson process. The other part of that is that those changes tend to be discrete in nature, that it's not a continuous type of slow process. It's like there's a bunch of jiggling around and then bang, there's the change. And the reason why we have confidence that these things are modeled as Poisson processes is because modeling them as such has been for really over a century of quantitative chemistry exceedingly effective in modeling experiments. And so we can then think about maybe what is our simplest model for this. Our simplest model for microtubule catastrophe might be that there is a single biochemical process that happens to trigger catastrophe. Now, that seems like a vague question, but it's actually really not, because what it does is it helps our experimentalists who maybe are more interested in the details of the chemistry be like, okay, well, what am I looking for? Is it going to be a multi-step chemical process? Is it going to be a single chemical process? This is a really fundamental question, and that's something this experiment can get at. So that's going to be our first model. And so what I'm going to do now is I'm going to shift over to Notebook. And if you want to follow along, you can. I'm going to switch my screen share out of this. There we go. And into the Notebook, which is here. Okay. All right. You know what? I'm going to increase the font size a little bit on this, Alex. I think it might be easier to read. Okay. So if you want to follow along, you can. Now, there's Alex shared with you a link to a GitHub page for this. I probably should have had it up here. It was, you know what? I don't have it. He sent it to you. Maybe Alex, you can paste it in the chat real quick.
Yeah. I'll paste it again in the chat.
Yeah, and you can follow along in Colab if you want. And that's fine. Or there are also instructions in the README of that GitHub page on how to install locally, so you can follow along. There are a couple packages that are not commonly used. They're things that I wrote. There are sets of tools. Thank you for that link there, Alex. If you want to follow along, you can. I don't really think it's necessary to fully get what I'm going to be doing here. Maybe you might want to run the notebooks later, but I'm really just going to kind of walk through this analysis so you can sort of, what's more important is sort of how we construct our models as they pertain to our experiment. And then, I don't want to say it. I hate saying this. The rest is just sampling, because there's a lot in that just sampling part. We don't want to trivialize that, but I really want to try to focus on sort of the approach that we take to the modeling. Okay, so I've already run all the cells, so I'm not going to rerun them. If there is a cell that's going to take a while to run, I'll let you know. So those of you who are following on Colab, you definitely want to run the first cell of this because that's going to take quite a few minutes to run because it's going to actually install Stan. You have to, each time you fire up a new Colab instance, you do have to install the packages that you need to get things done. And we're going to be using Stan as our modeling platform. Okay, so let's, the first thing that we're going to do, and I think pretty much in any analysis, is going to be exploratory data analysis. And so the data set is available in the Git repository, and it's actually so simple. I don't even need to use pandas or anything to load it in. I can just load it in straight as a NumPy array. It's just really, like I said, it's just a list of times for microtubule catastrophe, and it's something like 250, 300 some data points, something like that. And so,
and I have two questions for you, if
okay.
Justin. So one, wait, my mic is not here. That's better.
Yeah, I see the questions, and I'll address them. Okay. So the first question is,
I will repeat them because for the recording, people will not see the question. So
yeah, no, I was going to repeat it too. Okay.
Okay, perfect.
So all right. The first question is the time to catastrophe is the data set time to catastrophe or length of microtubule? Rate of growth seems quite constant. That is true. That's a good question. We saw in those chimegraphs that's what those plots are that I showed you, where the time is the vertical axis. That they look like perfectly sloped triangles, and that's because the growth is roughly constant. But remember, we're really just looking at what is the time to catastrophe, because the catastrophe time that's really clear in the chimograph. It's like, bang, it's gone, right? But to get the growth, really what you'd have to do is you'd have to actually do some sort of regression on the length versus time, if you wanted to like connect those two. And remember that what we're trying to do here is we're modeling this as a Poisson process, and the story behind we're going to be using an exponential distribution, which is the story behind that distribution is the amount of time it takes for the arrival of a Poisson process is exponentially distributed. And so it's really the time that we're interested in our model and not the length. Now, there are some very interesting studies of length control of microtubules. In fact, I give a homework problem in one of my other classes where we look at length control that happens in the presence of other biochemical species. This experiment, all that stuff is stripped out, but when you have these other regulatory things that can actually sense length, then we do care about the length. And so that question might seem like a simple question, but it actually leads to some very subtle considerations when we think about connecting our model to how we interpret the experiment. Now, if we had these sort of length sensing proteins involved that can actually affect polymerization rates based on length, then we definitely want to start to consider length. But really what we're looking at here is what is the timing of a biochemical event modeled as a Poisson process. And so in that case, it's the time that really matters. And that's exactly the kind of question that I think you need to be asking when you're designing experiments. And remember, when you design an experiment, you should have a model in mind. And combining those with models, that is exactly the type of question you need to be thinking about. Another question was about Stan. I think that was, if folks were familiar with Stan, I can briefly tell you what Stan is when we...
Yeah, that was just me. I was just curious. So.
Oh, that was you asking that question. Oh, okay.
Yeah. No, there was another question from Dan. So that first one was from Scott. Thanks a lot, Scott.
Oh, there it is.
And
okay, yeah.
Dan is asking, do you observe the rate of expansion as the same between MTs, with the only key difference being the time to catastrophe? MTs being microtubules.
Yes, in fact, MT is the abbreviation that is used in the literature as well. So maybe you're a biologist, Dan. But the answer to that is that their growth rate in these experimental conditions really doesn't vary much from microtubule to microtubule. They put in the cool thing with doing this experiment in the setup they have, it's called in vitro, meaning that it's just, they localize these things on a slide. They just have the tubulin in there, and they have the buffer conditions that they need for the thing to grow. There's no other regulatory proteins or anything like that. They strip away all of that other sort of regulation of variability that also induces variability. And really all you have is just the simple polymerization, which proceeds at pretty much a constant rate. Okay.
Cool. Thanks a
All
lot,
right.
everybody, for all these questions.
Well, so I just want to mention that I love these questions because they're really getting at what I think is so important for building your models, which is the domain knowledge. You need to have a basic understanding of your system before you start just throwing models at it, right? So these are great questions. Okay. So, all right. So we loaded our data set. It's just a set of times, and the classic variable for time in all of physical and biological and chemical sciences is t. I know it's not the most descriptive variable name, but it's one of the ones that's so ubiquitous, we're just going to use it. And now what I'm going to do is I'm going to plot an ECDF, an empirical cumulative distribution function. It's really, in my view, a preferred way to plot univariate data, meaning we have one quantitative variable. And the way you interpret an ECDF is that it looks like the cumulative distribution function for whatever the generative process is, right? So for probability distribution, you have for a continuous random variable, you can have a probability density function, and then with the same amount of information, you can have the cumulative distribution function. The way you interpret this is, let's say I go to the y-axis here at 5 minutes. So this says that 0.4 or 40% of all of the observed catastrophe events happened at 5 minutes or less. So that's how these things are interpreted. And so it's a good way to visualize a distribution. So you can say that, okay, I can have 30 minutes catastrophe. I've got this like tail out here that says that roughly 5% of them were longer than 15 minutes. Okay, so this is what the data set looks like. And so maybe it's funny. Usually when I teach, I'm asking students questions, and I know it's a little bit harder to do in this framework. And so I've been resisting asking you guys a bunch of questions, but I'm going to ask you anyway. Maybe Alex can answer if he wants. I'm going to put him on the spot even more later. But our first model is that we're going to model the time to catastrophe as an exponential distribution, as being exponentially distributed. So the story behind an exponential distribution is that it's the amount of time you have to wait for a single Poisson process to arrive. In other words, what that model means is that we're assuming that there's a single biochemical process that's going to trigger catastrophe. Now, we're gonna use that model, but just by looking at the data and maybe what you know about the distribution, do you think that that's going to be a reasonable model for this? Is there any features in that? So I got to know.
Yeah.
And I'm happy to pause just for a moment to maybe hear a quick explanation as to why. It's way more fun to talk to students. That's why I teach anyway.
Yeah. I would say no also.
Yeah. What feature of this is leading you to the no? I got another vote for no. Yeah.
Yeah, let's go.
Well, I'll let you ponder. It does sound reasonable to me too. It definitely sounds reasonable. Actually, this is a nice point. Okay, so when you're doing modeling, you want to start with the simplest reasonable model, and this is the simplest reasonable model. We know that this is a biochemical process. We know from over a century of experience that biochemical processes are well modeled by Poisson distributions. No, I didn't mean to say that. Are well modeled by Poisson processes. So that means that the time at which it takes for something to occur is going to be modeled by an exponential distribution. So it's a totally reasonable model, and I agree with the comment there that it sounds reasonable. But when we look at the data, and I don't know, as I'm sharing my screen, I see that my mouse is not visible, and there it is. Thank you. It says the data has an inflection point. That's the key, because that is not consistent with an exponential model. That's right. For an exponential, you will not have an inflection point. And so when you look at the CDF of an exponential distribution, there is no inflection point, and this ECDF has one. And so it's unlikely that this is going to be a good model. And you can tell that right away from the EDA. But we're going to start with exponential anyway, and so let's proceed with some modeling. All right. I love it.
Yeah,
good job.
Yeah.
Good stuff, everybody. Okay.
Yeah. And to make it clear, the inflection point you're talking about here is around 10, 15 minutes, right? Something like that.
No, the inflection point is around, looks like it's around 4 minutes. Because if you look on the left part of it, it's a convex function, and then it becomes a concave function to the right of that. Yeah.
It's a good thing that I asked.
And that is exactly right, Matt. We're talking about the exponential as a likelihood, and let's build the model. Okay. So here's our model 1, exponential. And I'm going to skip all this text. That's for you all to read. And so, so it's an exponential likelihood. So I'm going to scroll back for a second. Ignore that I just did that. So what I'm going to do is I'm actually a little bit old school. And I'm gonna share a weird screen here. A lot of people write on tablets, not me. Wait, where is it? There it is. Okay, I'm gonna write on paper. All right, so you can all see this? And here's my hand and my pen.
Yes.
All right. So our likelihood is going to be that our time to catastrophe is going to be exponentially distributed. Is that big enough? Can you guys see that? Should I write bigger, or can you see that okay?
Yeah, I think it's good.
And the exponential distribution is typically parameterized by a single parameter, which I'll call beta, the so-called rate at which the Poisson process arrives. And I'm going to assume IID. Now, not a lot of, in most of the literature, people don't write it this way. So I'm saying that time point measurement I is exponentially distributed, and that all of them are exponentially distributed. I always do this just to keep myself out of trouble, because to me it's explicitly saying that it's IID, identically and independent and identically distributed, right? Okay, now that's my likelihood. And so when you're doing modeling, right, you start with a likelihood that tells you how the data are generated. And that then tells you what are the parameters that we're going to try to estimate in our posterior. And in order to get to complete our model, we need to provide prior distribution for the parameters that parameterize the likelihood, right? And so what parameters are the likelihood here? This rate, beta. Now, it turns out that, I don't know about you, but for me, when I'm thinking about Poisson processes and rates and things, it's easier for me, instead of thinking about a rate, to think about a time scale. So I'm going to define by tau to be the inverse of the rate, because a rate has units of inverse time. Tau, right, has units of time. And so this is the characteristic time that I have to wait. It doesn't matter which one you choose. I just think for thinking about priors, it's easier for me to think about a time scale as opposed to a rate. So this is one little trick I want to share with you, which is choose a parameterization, at least for your priors, that is easiest for you to reason about when you're coming up with a prior. All right, so now we have to come up with a prior distribution for this time constant tau. All right. Oh, man. Well, we already saw some data, so we already know it's going to be roughly 15 minutes or something like that, right? But let's divorce ourselves from that and try to come up with a prior before we ever do the experiment. And so I'm going to demonstrate a way in which I like to come up with priors. I call it the bet the farm approach. But I'm going to demonstrate how you go about doing it with what might seem like something that's unrelated in a minute. And this is where I get to put Alex on the spot. Mm-hmm. All right. So, Alex, you and I have not discussed the question I'm about to ask you. I did tell you the other day that I'm going to put you on the spot. I'm going to ask you something that you probably don't know about to demonstrate that you actually know more about it than you think you do. So here we go. Alex, do you know what E. coli is?
Yes.
Right. So it's a bacterium.
I've heard of it.
That's right.
I could not recognize it in the street. But I've heard of it.
It'd be hard to see in the street, and I'm sure there's plenty of it in the street. But it's a bacterium that inhabits the guts of mammals. Okay, so my question for you is, how big, like how long, is an E. coli cell? A single E. coli cell.
I'd say definitely under 1 centimeter, and I don't know what the smallest cell can be, but let's say more than 1 micrometer. I think it's pretty wide prior already.
Okay. So I like what Alex did there, and I think so what I'm going to do now is I'm actually going to step through what happened in his mind when he was doing that. I'm going to pretend like I could just read his mind right there.
Yeah, how do you do that?
Yeah, okay. So here's how we're going to do it. So here's what I call the bet the farm approach. So there's a figure of speech. I don't know where everybody is in the world or what your language that you native language that you speak is, but there's a figure of speech in English called bet the farm, which means would you bet everything that you have? Right? Because in the old days, the farm was everything. It was you owned the farm. It produced your food. It provided. That's where your shelter was. The farm is all you have in this world. And so it was commonly used as, do you think, you would say, do I think LAFC is gonna win tomorrow night in the Leagues Cup quarterfinal? I do, but I wouldn't bet the farm, right? So meaning, yes, I think they will, but it's not a sure thing. Okay. So the bet the farm approach, I want you to take it literally. I want it to be like, would you be willing to sacrifice pretty much everything that you have on a bet? And here's the bet. The first bet that I'm going to ask you is, would you be willing to bet the farm, Alex, that an E. coli cell is shorter, not like smaller than a kilometer?
Yes.
No hesitation. He absolutely would. How about smaller than a meter?
Yes.
That's the length of your arm. I'm pretty sure the bacterium's not bigger than that, right? Okay. How about a tenth of a meter? Would you bet the farm on that? So that's going to be 10 centimeters.
Yeah, it's 1 decameter.
10 centimeters. Yeah, that's like the length of your finger.
Yeah. No, I would say I would bet it's below that. Yeah.
I think there's still no hesitation there. Would you bet the farm that it's below a centimeter?
That no. Because I don't know if E. coli can be adult or I don't know if E. coli grow increases or not, so I would not bet the farm.
Exactly. Okay. So he started to feel hesitation. Like as soon as you feel hesitation, that means you're not going to bet the farm. Okay. So our upper bound, so I'm going to write it down here. We have 1 centimeter. Yep. So that's our upper bound. Okay. Now I'm just going to tell you a little quick fact. The diameter, you might not know this, diameter of a hydrogen atom is about an angstrom. That's 10 to the minus 10 meters. Diameter across, it's 10 to the minus 11. It's a tenth of an angstrom. Diameter across a DNA molecule, not the length, just the diameter of DNA, is about 2 nanometers. All right. So here's the question. Would you bet the farm that the length of an E. coli cell is bigger than a nanometer?
Yes.
No hesitation. So it's bigger than 10 to the minus 9 meters. So I should say 1 centimeter. This is equal to 10 to the minus 2 meters. Okay. So right now you're at 10 to the minus 9 on the lower end, and you bet the farm on that. Okay. How about 10 nanometers? Would you bet the farm that it's bigger than 10 nanometers?
So 10 to the minus 8?
Yeah.
Yeah.
Okay. I didn't feel much hesitation there. Okay. How about 10 to the minus 7 nanometers?
Also, yeah.
Okay. So you're absolutely sure that it's bigger than 10 to the minus 7 nanometers. So that's a tenth of a micron. Okay. All right. How about a micron?
Yeah. No, here I would not be sure.
He's uncomfortable. So micron. Okay. So he has 1 micrometer. So this is 10 to the minus 6 meters. right?
Yeah.
Okay. Now, one of the things that to get your then estimate is you'll take the geometric mean of these two things.
Mm-hmm.
And so Alex says that E. coli is going to be about, so the mean of 10 to the minus 2 and 10 to the minus 6, the geometric mean of that, right? That's going to be 10 to the minus 4.
Mm-hmm.
Okay. So he's going to say that it's 10 to the minus 4 meters or 100 microns.
Mm-hmm.
Okay. Well, it turns out that that's about the size of your cells, your bigger cells. So a human egg is roughly 500 microns. That's one of the larger cells. You definitely have neurons that are 100 microns. You'll have some muscle cells that are that big. E. coli is actually quite a bit smaller. It's actually about 1 micron.
Okay, so
but
quite smaller, like by a factor
quite a
of
bit
100.
smaller. Yes, by a factor of 100. But as these things go, that's it's a factor of 100, but that's 2 orders of magnitude. That's not that bad. Now, so now if I have these bounds, so here is, I'm going to do log base 10 of the length. And so you're going from minus 6 to minus 2, right? Now, we might just sketch that it might probably look something like this, right? Because if I'm putting bounds on the end of that, then this is going to be, I would think that most of my probability mass would sort of lie between these. And then most likely it's not where I think my bound is. Like, I'm uncomfortable betting the farm, but I'd still probably bet that it's bigger than minus 6, or I'd still probably bet that it's less than a centimeter, right? And so then what I'll do is I'll say, okay, well, I'm going to say that this is normally distributed. And what I'll do is I'll say that its mean is going to be, in this case, minus 4, right? And then I'll do it so that I'm 2 standard deviations out to get to my bounds. So that means that I'm going to have 95% of my probability mass between my bounds on a log scale. Yeah. So in this case, that would mean I'd have a standard deviation of 1, because if I go 2 standard deviations that way, that's and then 2 standard, that's 95%. Yeah. So standard deviation of 1. Mm-hmm. And so what this tells, if I were to write this down, I would say that log base 10 of the length of E. coli is going to be normally distributed minus 4 and 1. Okay, so we're going to take a bet the farm approach to the time it takes for microtubule catastrophe. And so let's do that. And maybe I'll just do that with you, Alex, even though you're not a biologist. Do you want to do it again, or I can do it myself? Sure. It's up to you. You want to try it again?
Yeah.
Okay. Sure. Okay.
It's always fun to choose priors.
All right. So let's go for the upper bound. All right. Would you? Alex is becoming a biologist. Would you bet the farm that the time it takes for microtubule catastrophe is less than a year?
Yeah.
Okay. A month?
Yeah.
A day?
Mm-hmm.
Okay. Let's go about an hour.
No.
There was a hesitation there. Mm-hmm. So an hour. So let's do it here. So we have Alex's tau. So the upper bound is 1 hour. And just to make the units consistent, let's we'll come back to it. We'll make the units because our time's always weird, because for some dumb reason we decided to have 60 seconds and blah blah.
Yeah.
Okay. Yeah. Right. Okay. It was easier with the lengths, wasn't it? All right. Yeah. So let's go. And for you, it was really just kind of gut feel. But maybe can you tell me why you felt uncomfortable at the hour mark?
Because we saw the graphs with about 15 minutes, and I don't know if those experiments we saw were kind of normal. And if they were, that means maybe sometimes there can be very, very long microtubules length that can be like, I don't know, go up to 1 hour. And like 1 hour is really my upper bound. I really think that would be like at the very, like the like you did with the normal distribution, that would be the right at the right of the distribution. Yeah.
Very good. Right. So now that was based on data that we saw that we're actually doing the model of. So it'd be nice if we could have a prior independent of having observed the data. Because ideally what you want to do is you want to come up with your model before you even do the experiment.
Yeah.
You want, as Darwin said, any observation, if it's going to be worth anything, should be for or against some observation. So really, it should be in the experimental design phase that you already have your priors in mind. So I showed you the beautiful movie of the cell division of the embryo, right?
Yeah.
And there was a little timestamp on that. And if you noticed, that took on the order of minutes.
Yes.
Like 10-ish minutes, right? And so if you think about that, okay, the dynamics of the microtubules are what's powering all of that. Now there's also regulatory proteins involved. So, and that cell division happens in the order of minutes. Okay, maybe order of an hour would be kind of another order of magnitude beyond that.
Yes.
And so that's kind of how I came up with mine. It's funny, like the things that you see, it's called, there's a wonderful book by Sanjoy Mahajan. I have it. I won't, wait, where is it? Actually, I think it's right here. It's in reach, I think. Let me see, where is it? Yeah, here it is, right here. I've got it. So, whoops, here. It's called Street Fighting Mathematics, the Art of Educated Guessing and Opportunistic Problem Solving. This is a wonderful book, and it sort of talks to you about, how you can street fight your way, meaning how you can really help, draw on what your actual experiences are to get to a prior. He doesn't actually talk specifically about priors, but it's the same idea. Okay, so let's go from the lower bound. How about, let's see. Let's go for a lower bound. So let's talk about, let's see what a good starting point is. Let's talk with try a nanosecond.
So it's 10 to the minus 9.
Yeah, 10 to the minus 9 seconds.
No, I think it's bigger. Okay. It takes more time.
Okay, microsecond.
So it's 10 to the minus 3.
Micro 6, 10 to the minus 6 seconds.
Oh yeah, no. Here I would say that's the lower bound. 10 minus 6.
Okay, how about 10 to the minus 3? That'd be a millisecond.
Yeah. No, I mean, I would say that it doesn't take that 10 to the minus 6. No, 10 to the minus 3 is the lower bound. Okay,
so you start to feel uncomfortable. Sure. Okay, all right. So now that your lower bound is 10 to the minus 3 seconds. All right, so your upper bound is 3,600 seconds, 10 to the minus 3 seconds. So that's basically 10 to the minus 3 to 10 to the minus 3.
Yes.
That's 10 to the minus 3 to 10 to the third, roughly.
Yeah.
Yep. Cool. All right. My bound was a little bit higher on the other end, and the reason I know that is because mitotic spindles are stable for a longer time period. They don't undergo catastrophe as rapidly, but I happen to know a little bit more. And so that's what happens. If you have more background information, your priors might be a little bit narrower. Yeah. So what we just saw is that this technique of bet the farm has actually properly codified our respective prior knowledges. I happen to have studied cell biology a bit more than you. My priors are a little bit narrower. Not much, actually, but a little bit narrower. And so this is exactly, I think, a really great way to go about specifying priors.
Yeah. No, for sure.
So
I always tell that also to students who are like, I don't know anything. I'm not sure you don't know anything. And you can use these kind of methods to show that you know more than you think. It's just that you've forgot that you knew more in a way.
So there's a question right here. It says, is there a futile cycle early on, like a polymerization of 1, where the failure to extend is quite high, as opposed to a more processive extension phase? If so, could this define the lower bound of the prior? That's an actually very interesting biochemistry question. That is true. the initiation of polymerization is itself it's actually almost a qualitatively different biochemical process than extension. This is part of the beauty of this experiment that Melissa and Maria put together, which was they bypassed that technicality by stabilizing that red part of the microtubule. So all of this is a great point right there that was made in the chat. They thought of that. That's why they built the experiment the way they did, to try to avoid that. But you're right, that is true. Initiation of polymerization is it's like failure, and then finally it goes. It's totally different. It's a different process, and they bypass that by experimental design. I always tell my students, like, you can model your way around stuff as much as you want, but really what you should be doing is spending your time designing your experiment to get all that confounding stuff that you think you can model your way around. Get it out of the experiment. Design your experiment well. That's where you should really put a lot of your effort. Now, the modeling and design is a cycle, right? And I'm talking even before you do the experiment. You come up with a model for what you think is going to happen, and you need to make sure that all of the stuff that you're assuming in the model that you can actually realize in your experiment, right? And especially including these sort of difficult things to model that could really be present in the in an experiment if you don't correct for it. Maria and Melissa are awesome scientists, if you haven't already gathered. Okay, so I'm gonna go back to the notebook, and if we don't get through all this, I'm fine with that, because I think that these lessons about thinking about priors and how you go about modeling are just so essential, and that I really don't mind having time and discussion on them. And I think your guys' questions are great, so I'm happy to keep doing that. All right, so this just kind of goes through the bet the farm approach that I just worked out, and then here is now my full model. Oh, I just realized you can't see my mouse. Okay, so we're looking at the equations now. These four expressions toward the top of the window, and let's start at the bottom, because that's the likelihood, and that I already wrote down. So that's the exponential likelihood. Now it's parameterized by beta. Now why? Now we just reasoned about tau. Why would I parameterize this by beta? And here's another tip/trick of doing modeling. Throughout all of your work, I think for the way you actually do it, use a distribution, always use the same parameterization. Decide on a set of parameterization for your distributions and use it. What I do is a parameterization that is used by default in Stan. So I choose Stan's parameterization, and that's what I use. Now, you may choose to use PyMC's parameterization, which I think is not the same as Stan's, but it's not that dissimilar. It's the same as I think SciPy's or NumPy's. I don't remember which one.
Yeah, but usually we use the same parameterizations as NumPy.
NumPy. Usually. That would make more sense. Yeah.
Depends, but
the SciPy ones are a little esoteric, I think, actually. But the NumPy ones are pretty good. But those are not exactly the same as Stan. I chose Stan, and I use everything to match Stan. And so that's why I'm always gonna write, when I actually write the distribution down, it's always going to be with the same parameterization, and I'm going to do transformations separate from that. And it just keeps me out of trouble, right? And so what I'm going to do is I'm going to use beta, but I know that I'm going to be reasoning my prior using tau. So I'm going to have this transformation. And furthermore, I didn't reason about tau. I reasoned about the log base 10 of tau. Or I should say Alex did. And he said it's going to go from 10 to the minus 3 to 10 to the third. And so now these two things together constitute a log normal distribution. Now, have any of you used the log normal distribution? The answer is probably yes. It's fairly widely used. And I think more times than not, I screw it up. I screw up the log normal every time.
Yeah.
Not every time, 80% of the time, I screw it up. And so I don't do it anymore.
Yeah.
I always reason about the log base 10 of something with a straight up normal, and then I do the transformation. It keeps me out of trouble.
Yeah, same.
Yep. because I'll screw. If I use log normal, it's over.
Yeah.
So it's hours of debugging if I do that.
Yeah.
So yeah. So I'm going to say, okay, for my prior is a little different. So my mu and sigma are going to be different than Alex's. His mu would be 0 because he's going from -3 to 3, and then his standard deviation is going to be 1.5. Mine's a little bit tighter and a little bit shifted to the right because I had the hour as my upper bound, but my lower bound was a little bit further to the right. So I have a 1/2 and 3/4, but it's pretty close to what Alex had.
Yeah.
Okay, so this is my model. That's it. Oh wow, we're an hour in. Okay, so what I'm going to do is I'm going to skip the modeling. I'm sorry. I'm going to talk only about the modeling. I'm going to skip the implementation. Many of you are PyMC users. I use Stan. It doesn't matter. We're
It's going to be
putting
similar
in this model.
Yeah.
Pardon me?
Yeah, it's going to be similar to implement it.
Yeah.
So it's actually
Absolutely
like having the model and talking through it, then implementing it is a bit different, but not that different.
That's right. Okay, so that's cool in the gang. And so now what I'm going to do is I'm going to scroll all the way down, do prior predictive checks, blah, 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 stand. Okay. Then what I like to do to check my models is I like to make a plot like this. And so I'm going to explain what this is. In orange is the ECDF of the actual data. So I already plotted this. I plotted it with dots before. The axis, the data stopped at around 30 minutes. And so if I want to zoom in on that part, I can. So in orange is the ECDF. It's like the staircase style of the ECDF. So that's what my actual data look like. Okay. So now what I did is I actually implemented this model in Stan. And so I did a parameter. I sampled out of the posterior for this parameter beta. And so I got posterior samples for that. And then what I did is I generated data sets using the posterior parameterization. Okay, so for example, let's give you an example of how this works. For a given beta, from a posterior sample of beta, I then plug that into my likelihood, exponential parameterized by beta, and I generate the same number of time points as I got from my actual experiment.
This is also called posterior predictive checks.
That's right. That's exactly what that is.
So if you're using PyMC, that's when you use pm.sample posterior predictive, and PyMC does that automatically. And if you come from classic ML world, I think in that world, I actually like that name. I think it's better. It's called in-sample predictions. And so that's useful. I think it's a useful name. So you probably have heard about that already.
Exactly. That's exactly what I'm doing. I'm just saying for every posterior sample of beta, I'm going to use that beta and my exponential likelihood. I'm going to I'm going to produce a new data set. And I do that over and over again. For every posterior sample, I took 8,000 samples, I think, or 4,000 samples. It doesn't matter. And then what I did here is I plotted the ECDF of my samples. That's what the dark blue line is. And you can't see my mouse. The dark blue line is the ECDF of the samples. Okay. Now, the faintest line, 95% of the ECDF of those posterior predictive samples that I draw lie within that faint line. Then the middle degree of blue shading, that's 68% of my posterior predictive samples for the ECDF lie within there. So basically for each data set that I generate, for each posterior sample beta, I generate a data set and I compute its ECDF of that data set. I do that again. For each beta, generate the ECDF. Do it over and over and over again. And then I have this envelope. And now if I look at this, the blue tells me what types of data sets could be generated from this model. The orange tells me what type of data set was generated by nature. nature does not seem to be generating the same types of data sets that the model generates. And so your intuition at the beginning is right, that the exponential distribution is just not going to cut it.
We really see the inflection point now with the
Here's the data.
Yeah.
And now one of the ways I like to plot this actually is what I'll do, because sometimes it's harder to see because these blue areas can be kind of narrow. What I'll do is I'll take that dark blue line and I'll set that to be 0. And then everything else is the difference off of that. And I'll show you what that looks like. So if I do that, that's down here, right? So that's blue now. That is my median ECDF of all of my samples, of my posterior predictive samples. The darker blue envelope contains 68% of the samples as a difference from the median. The light blue is 95 as a difference from the median. And then the orange is my data set as a difference from the median. Yeah. And now you can really see the differences.
Yeah.
And as you can see at the bottom, it says it fails terribly. Okay. That's great. Now, that model didn't work. So we got to come up with another model. So here's another tip and trick on how we do this. When you're coming up with your next model, a good practice is the following. We had a model. We had the simplest possible plausible explanation for what our data might look like, and it failed. And so now we need to add flexibility, which really comes through complexity, to the model. And as we're doing it, we don't want to just come up with a model that's totally unmoored from the previous one we came up with. We want to add that complexity systematically, which means that we really want our more complex model to be either a special case or we want to have our simpler model to be either a special case or a limit of our more complex model. All right. So what might be next? Well, the next obvious thing we might consider is, look, we modeled this thing as a single Poisson process arriving. Maybe it takes more than one chemical process to trigger this catastrophe. And that right there is already a scientific advancement if we can show that that's the case. That we know that it's not as simple as just one biochemical process triggers it, that there's got to be more than one. And so our next model, we might choose the gamma distribution. Now, maybe you're not familiar with the gamma distribution, so I'll show you a nice tool that I use as I think about these distributions. So this is the Probability Distribution Explorer. It's a website. It's distributionexplorer.github.io. It was developed by me, and I just built it for my students, and it turns out that a lot of people actually use it. And so you can click on these different name distributions. So we click on the gamma. It tells us the story of the distribution. In particular, it tells us that it's the amount of time we have to wait for alpha arrivals of a Poisson process. And importantly, what it gives us is actually, if you scroll down here, it'll give you the PDF and CDF, and you can see how they vary based on the respective parameters. And so I can mess with alpha and beta. This is also useful, by the way, if you're trying to come up with priors for things. I often, if I choose a distribution for prior, I can sort of mess with the plot to have it match my what I think about what the prior distribution should look like if I were to sketch it. But we're not going to use it for that purpose here. Really, what we're going to look at is we're going to say, okay, well, this is the gamma distribution. And if we look at its CDF, right, so if we look over here, we see that unlike the exponential, it does actually have an inflection point. And this makes sense, right? You have to wait for more than one arrival. So you're not going to, the most probable arrival time for the exponential is immediately. But if I got to wait for more than one, then I'm going to have to wait a little bit longer. And that's why I'm going to have on the left end, it's going to go to, be close to 0 for a while until I wait long enough for both Poisson processes to arrive. So I'm going to use the gamma distribution. And importantly, if I look at related distributions, it says right here that a special case where alpha equals 1, this is the middle bullet point under related distributions, the special case where alpha equals 1 is an exponential distribution. So that means that, okay, now I'm adding complexity. It's got two parameters, alpha and beta. Beta is still the rate of the Poisson process arriving. And actually I say what the parameters are here. I say the number of arrivals alpha and the rate of arrivals beta. And so we can really use the same prior for beta, right? Because it's the same idea. It's the exact same idea. So we use the same prior. Now, alpha is the number of arrivals. Now, one of the things I'll just point out briefly that's useful with the Distribution Explorer, which also helps keep me out of trouble, is it actually, everything in Distribution Explorer is parameterized according to what Stan uses. And then it actually tells you to match what Stan's parameterization is, what you would need to do in NumPy and SciPy. I also have a pull request for that's been completed for Julia, and R will be added here soon. I have a collaborator who's going to be adding R as well, so you can look for that in the future.
Nice.
Okay.
Really cool. Yeah, well done on developing that tool.
Thank you. This saves me
really
so much time with these parameterizations.
Yeah.
What's the worst? What's the most notoriously difficult parameterized distribution, do you think, Alex?
Gamma is really hard. Most of the time.
It is.
Yeah. Gamma.
For me, it's negative binomial. That one wrecks me every time.
Also, I hate the name negative binomial because
I hate
it's
it
more
too.
like a gamma Poisson, and for some reason we call that negative binomial, and then people get confused, me including, with beta binomial, and it's just awful.
So as I put here, for this reason, the negative binomial distribution is sometimes called the gamma Poisson distribution. I mean, this one is a mess. And this Distribution Explorer has saved me so much time with this thing. So this is just a useful tool. I think that might be able to help you. Okay.
Yeah, it's really useful. And then you can use that. And then in combination, if you're using Python, so in PyMC, we already have the pm.find constraint prior function that can really help you. So it's like, if you know you're using a gamma, you can tell PyMC, okay, so give me a gamma which has, 95% of its math, for instance, between 1 and 3. And then PyMC will optimize the distribution and then gives you back the parameters that you should use as a prior. And you can use that directly in a model, for instance, in PyMC, so that you don't even have to write the parameters. And so that's the first, let's say, raw thing that you can do, which is already super useful in combination with your Explorer. And
I've actually started hacking it together. That's going to be added to the Explorer, and it'll all be done with JavaScript. So it'll all be native in the
perfect.
So that's a project I've started working on.
It's interesting, but it's really hard math under the
hood. It is.
Once
you have to derive it for each one.
Once you start looking into that, you're like, oh my god, that's way more math than I thought.
That's right. And then try doing it in JavaScript. But anyway, so that's what I'm doing.
But I mean, if it's useful to you, I could put you in contact with the folks who are doing Prelease. So Prelease is a Python package. Right now, it's you can pip install it. It's not yet on Conda and Mamba, I think, but you can pip install it. And basically, Prelease is even more advanced way of choosing priors that way, where you specify the constraints and the distribution that you want, and then the tool gives you the good parameter, the corresponding parameters to your constraint, because in a way you don't really care about the parameters of the prior, you care about the shape of the distribution and what it implies for your model. So Prelease is an even more advanced and really at the scientific frontier of doing that. It comes from Osvaldo Martin and other folks who worked on that at Aalto University with Aki Vettari.
I know that crew.
Exactly.
There's a nice question
I
here.
put the link here.
Great, excellent. There's a nice question here that says we jumped into picking and testing likelihood distributions for time to catastrophe, but what would this look like as a directed acyclic graph? T to catastrophe. Well, your observation is a time to catastrophe, and as a directed acyclic graph, it's the challenge is that if you're trying to be causal, you simply don't know what the biochemical processes are. And so it's really just you have the catastrophe happens, and that is there one process that leads to it. And so the next question is, why not consider precursor concentration? I think by that you mean tubulin concentration, microtubule length, et cetera, as well as age. So, right, another great question. The paper is linked to in the notebooks. They actually do it with various tubulin concentration, and they also do it. So they actually do that and investigate the dependence on that. The idea is that would change the rate parameter. Possibly, turns out it doesn't, which is very interesting. It polymerizes faster. You get longer microtubules, but the time it takes to catastrophe does not depend on the tubulin concentration, which tells you that your model, where you're saying, okay, well, it's just a Poisson process that triggers catastrophe, makes sense. It's independent of how much tubulin you have. And so, so let's, we only have like 10 more minutes. So let's proceed to the gamma distribution. So we can code that up and do all the rigmarole, right? And then here's its posterior predictive check. Now, again,
I mean, you
orange
can continue a bit more than 10 minutes.
I'm happy to. And I know some of you may have places you need to go, whatever. I will not feel at all offended if you need to leave. So
you will have access to the recordings anyway.
Right. Okay. Although I'd love you to stay. This has been really fun. Okay. So the gamma model, what we have is, look, this is the data set. You can't see my mouse, but the orange is the data set, and it's all within the envelope of 95%, except roughly 5% of it, which we would expect, actually. And so this actually does pass the posterior predictive check. It says, okay, well, look, the data set that we observed could be generated by this model, parameterized by the parameter values that are highly probable in the posterior distribution. So that's good. This model holds muster. And if we look at, I won't go into the Weibull, but if we then, this is what I call a corner plot. So the two parameters we have are alpha and beta. I like to think about 1 over beta. So we can always do a transformation to look at the tau. And we see that the characteristic rate at which these Poisson processes arrive is about 2.2 minutes, is about once every 2.2 minutes. So for 2 of them or 3 of them to arrive, it'd be somewhere between, on average, 10-ish, which is what we saw in the data, actually, just kind of looking at it, eyeballing it. And then if we look at the number of Poisson processes, alpha is right around 2.9-ish, but, it spreads between 2.6 and, say, 3.2. And so it's probably about 3 Poisson processes, right? So we can feel pretty good about this. And this is actually what Melissa and Maria did in their paper. And they reported they used a gamma distribution. And this is all fine and good. But I guess since we are short on time, but nonetheless, you all said you like the Socratic method. So let's continue with it. Does anybody see a problem with this? Look, everything seems to check out. Does anybody see a fundamental issue with using a gamma distribution like this?
Can you remind what alpha is here on the x-axis?
That's a great, yes, very, very important. The story behind the gamma distribution is that you have a Poisson process, and the amount of time it takes for alpha arrivals of that Poisson process to arrive is gamma distributed, okay? So the rate of the Poisson process is beta, and you're waiting for alpha arrivals. Now, alpha can be non-integer. Mm-hmm. So it kind of murks up it makes the story a little bit more murky, but alpha can be non-integer. And so what we're seeing is that the most probable alpha is somewhere around like 2.9.
Yeah.
So it's about 3.
And so there is a negative correlation between alpha, which is the gamma rate, and tau, which is the exponential rate. Does that make sense?
Tau is a time scale for the arrival of the Poisson process, yeah. And so in the
does that
posterior,
make
there
sense
is,
biologically?
well, it makes sense inferentially, because if you think about it, if I'm saying that if I have to wait for fewer arrivals, right, then the amount of time it would take per arrival should be longer, because I'm still observing the time that,
The same time.
I wait.
Yeah.
Yeah, I wait some amount of
Yeah.
time and I make an observation. If
Yeah.
I have to wait for less arrivals, each one should take longer. So I do expect there to be a negative correlation there. That makes total sense.
Yeah. So
that's
good.
not a problem.
Yeah.
Yeah.
Oh, that's good.
Yeah.
But yeah, that means, does that mean, so that's the posterior, right? That's once you sampled. Okay.
Yeah.
Do we have any divergences or things like that? Because it can be...
you can go in the notebook. I have all of the diagnostics. Everything checks out. It's beautiful.
Nice. Oh, yeah. Yeah, because it can be challenging if there's some high correlation like that for MCMC.
Yep.
That's cool. Okay, so we have the correlation. It's good, and that's what we want.
So sampling's good. Agreement with the data are good. Here's my problem with it. If I'm talking about biochemical processes, they do not arrive at the same rate. The rate of a phosphorylation is going to be different than the rate of a methylation. Whatever my chemical process is, by a given enzyme, or like they're highly variable. In fact, enzymatic catalysis rates can vary, and I'm not making this up, over 30 orders of magnitude in speed.
Oh, wow. Okay.
So the classic example is the degradation of cellulose, right? So, Alex, you're from France. There are buildings there that are hundreds of years old and made out of wood.
Yeah.
Right? And yet, termites can break down cellulose in seconds, right? And so that the rate at which you have this chemical reaction of breaking down cellulose can vary over huge amounts of time. And so what we have here is we've modeled this where it's a non-integer number of arrivals of a Poisson process at the same rate. Yeah. And it just doesn't jibe with how biochemistry works. And
so
this
tau
I want to
should
emphasize.
Be allowed to change. You're saying tau should be allowed to vary according to the process.
Yes, and they should be discrete. This is another thing. Throughout the course of over the last well over 100 years, modeling chemical events as discrete Poisson processes has been wildly successful. And so we really should model this as discrete Poisson processes that have to arrive, perhaps in succession, but with different rates. Okay, so since you're letting me going to go over, I'm going to show you where frequentism fails. That's always fun for we Bayesian folks, right?
Oh, yeah. I mean, you're good here because people are going to stay with that cliffhanger.
Yeah, exactly. All right. So here's what I'm going to do. I'm going to say, you know what? This gamma thing's all fine and good, but I actually I'm going to have to build my own model. I'm going to have to come up with a story and then derive the probability distribution for the likelihood. And I was going to do this, but this is the integral I told you I was going to perform live, but I'm not going to do it. I'm just going to state the result. So what I'm going to do is instead I'm going to say, okay, I want to derive a probability distribution for the amount of time it takes for the arrival of, let's say, 2 Poisson processes in succession. And these Poisson processes have different rates. So what you're modeling there is that there are 2 biochemical events that have to happen in succession, but they're different biochemical events. And this is biologically speaking, this is a much more plausible model. The gamma doesn't even make sense because you have non-integer number of arrivals of events, and they're all at the same rate somehow, right? So I'm actually gonna have to build my own model. And that's another point I want to make to you, which is that sometimes the off-the-shelf distributions just simply aren't what you need. You have to derive your own. And that usually involves some math, some calculus, and et cetera. So in the second notebook, this is catastrophe 2. What we do is we have this 2-step model. And it turns out that we can derive, and I was going to do this on paper, but in the interest of time, I won't, that this would be the likelihood for the arrival of 2 Poisson processes where the characteristic timescale of arrival for the first one is tau 1, The characteristic arrival time for the second one is tau 2. And then this is my likelihood for that. All right. So I've identified my parameters. I got my likelihood. It's tau 1 and tau 2. Or conversely, I could look at their inverse, which is beta 1 and beta 2. Oh, I love this question. Oh, such a question. Can a multi-stage process be reduced to a single rate constant for the rate limiting step? Yes, and that's going to be the last point I make. And that is brilliant. That's such a great question. That is exactly what we should be thinking. My guess is that Dan, who asked that question, has experience as a chemist and thinks about this term rate limiting step. And that is exactly what is going to evolve directly from our analysis, is we're going to see a rate limiting step staring us right in the face. Okay. But first, let's see what happens if we take a frequentist approach. This is fun. Okay. So I'm going to share. I got to switch share here. I know we're going over, guys. I'm sorry. I'm having too much fun. This is really great. Okay. So here we are. So check this out. So here's what I did. I did a maximum likelihood estimate. And the maximum likelihood estimate I got was actually, and I did it in terms of beta 1 and beta 2. These are the inverse of the taus. It doesn't matter. You can do tau 1, tau 2, or beta 1, beta 2. It doesn't matter. So I did the inverse, beta 1, beta 2. Okay. So this is the maximum likelihood estimate right here, this open circle. And notice that it ends up having beta 1 and beta 2 being equal at about 0.315 inverse minutes, which is roughly the rate we would get. The inverse of that is approximately the 7 or 8 minutes total or something like that. Okay. So that's my MLE, and it turns out that they have exactly the same rate, which makes this a special case of a gamma distribution with alpha equals identically 2, right? Because if I have two Poisson processes that arrive in succession, but they arrive at the same rate, that's the same story as a gamma distribution. Okay. So now what I did is I did a bootstrap. So I resampled the data and then I redid the MLE, because my idea is I want to get a confidence interval on the maximum likelihood estimate for these parameters, or more specifically a confidence region. So this is a plot of the confidence region, because each one of these points is a bootstrap replicate of the maximum likelihood estimate for those parameters beta 1 and beta 2. And look at this. In 42% of the bootstrap replicates, beta 1 and beta 2 are equal to each other. In 19% of the bootstrap replicates, beta 2 is actually infinity, and beta 1, so I've colored this in red because I can't plot infinity on here. It just means that beta 2 is infinity, and then so you can see beta 1 ranges here. Now that is a special case of the exponential. So right, where one of the rates is infinity, which means it's instant, and then you just have to wait for one process. So that's the exponential limit. This is the gamma limit. And then 39% of the samples, the remaining, are these, which has beta 1 and beta 2 not equal to each other, which means that it's 2 steps in succession. So, my God, I mean, look at that. It's like, this is what frequentist gives us. It's a very curious confidence region. And I don't even really know what to say about that besides just kind of showing it. You can do a similar thing like a posterior predictive check to kind of see how well the CDFs match, and it'll fail there too. But this is very tricky. And I will say that you had to get this, if you just naively just throw it into an MLE algorithm, it will not work.
And is it because actually, there is no priors? And here, the fact that the parameters are really hard to identify, especially because they are very correlated. And if you don't have any priors, then that means it's really hard to come up with good estimates.
That's part of it. There's also this weird structure to the likelihood function. Again, this is not the Bayesian likelihood. This is the frequentist likelihood function. The problem is that when you maximize a likelihood, you're looking at a single you're looking at a single point estimate for the parameter values.
Yeah.
And that's what an MLE is.
Mm.
And the likelihood function itself has a weird shape to it, which is not captured if you just are reporting the maximum of the likelihood function. Whereas in the Bayesian context, you actually get to see the whole shape of your posterior distribution. You're not, we're actually looking at the entire distribution. If we just tried to refer to the maximum of the posterior distribution, or the so-called MAP, maximum a posteriori probability parameter values, that's fairly useless because it's just one point estimate for the parameters. What an MLE is fundamentally is it's a point estimate, and the way you can get a confidence interval or confidence region on it is by doing something like bootstrapping, or having in mind some generative model where you make some approximations, Gaussian approximations, or these sort of things. But we have plenty of data, so bootstrap works. The problem is that the thing that we're estimating, the maximum likelihood, is really uninterpretable. It doesn't even, right? It's convoluted anyway, right? So the Bayesian approach is nice because you're just looking at the posterior distribution of the parameters, and we can do that. And so I'm gonna share now the Bayesian one. Let's see. There it is. Okay. Same priors, same business. Okay. Prior prediction checks. Okay, I'm just going to scroll down. Okay. So here's, I'll show you the corner plot first. Okay. So this is what the corner plot looks like. And you can see that there's this weird sort of structure down here. And that's just because we're handling the symmetry. Because if you look at the expression for the likelihood, tau 1 and tau 2 are totally interchangeable. You get the same likelihood regardless of you interchange them. So if you actually just reflect this cloud across that line there, that's what it would be like. I've just corrected. It's just that we're only looking at sort of half the posterior, where we've defined tau 2 to be bigger than tau 1. That's all. And so this makes sense. It tells us that actually we have a fair amount of probability mass close to where they're equal to each other, but there's plenty of probability mass where they're unequal. And so the posterior predictive check, though, fails pretty spectacularly. Right? It's not two-step. And it stands to reason, right? When we did the gamma, we found that it was roughly 3. And so now if we go to the corner plot, we see, we have definitely 2 different time scales, right? This one's right around 3. This one's around 3.5. But we've restricted it to 2 steps. So what I'm going to do in the final analysis is instead, I'm going to take M steps. Now, this is a god-awful mess, but I'll just show you. I had to derive it, right? And sometimes you got to do this, okay? And I did it by doing a bunch of integrals, and it's fine. I did them, okay? So now let's say I have M Poisson processes that arrive with characteristic time tau 1, tau 2, tau 3, tau M. And now this is the model I'm going to use, and I have M parameters, all these taus, okay? And so this is an ugly expression. There's a weird product and a sum and all this blah, blah. But just so we can see it, I wrote it out for the first few M's. So there's your exponential for M equals 1. That's a single step. Here's the one we just saw for 2-step. Here's 3-step, 4-step, and so on. Okay. Now, coding this up in Stan, it's a bit of a mess. Whatever. I did it. You can read the notebook. What I want to show are the results. And diagnostics I'll check out. Oh, by the way, if you're going to, so I sampled M1 through 6. If you're going to run this on your own, on my machine, on my 2018 Mac Mini, it took 2 hours to do all of it for the sampling. It'll be slower on Colab, on the free tier, because you only have 2 cores, but yeah.
And how many data points?
How many data points? There's only like 300 in the data set. So the difficulty in the sampling is because the model's challenging.
Yeah, for sure.
But it's still a healthy. The sampler's all healthy. It's all good. Yep.
Yeah.
So All right, m equals 1. We already saw that. That fails. That's exponential. Here's m equal 2. That's the two-step. Fail. M equal 3. Beautiful. But wait a minute. 4. Beautiful. 5. Beautiful. 6. Beautiful. So these all seem to work. So 3 and above all work. So Scott asks overfitting, and I would say kind of. I think overfitting is a difficult term to parse sometimes. I prefer to try to be a little bit more specific in my case-specific way of the diagnostics. And here's how we're going to do it. I'm going to plot. So this is a little bit. I'm gonna shrink the image so I think you can still see the plots. Okay, it's still clear what I'm plotting, right? You can see that, okay, Alex? Okay. So what I'm plotting here is the ECDF of my samples for the given taus, right? And if we look at m equals 1, it says that my tau, and it looks funny because all the x-axes are linked. That's why, so we can make apples-to-apples comparison. Okay, so it says tau is around 6.5 minutes for exponential. That was bad anyway. This says that the taus are roughly, 3 and a little bit more than 3. We already saw that. But now let's look at m equals 3. This says that one of the taus is 2.5. The next tau is 2, and the next tau is about 1.7. Okay, so slow, faster, faster. Now look at m equals 4. These are roughly the same as the m equals 3 one. These first, you can't see my mouse, sorry. The 3 to the right are roughly the same as the m equals 3 one, right? But look at the one furthest to the left on m equals 4. That's very short times. And now look at m equals 5, another one that's even shorter. M equals 6, even shorter. And so, Scott, you're right. We're getting overfitting on models 4, 5, and 6. But what it's saying is that if you look at those are all very fast times. And they're actually at faster timescales than really what we're starting to observe for the catastrophe times. They're actually, some of them are faster timescales than what the frame rate of the movies they were taking was. So they couldn't even resolve catastrophes at that fine-grained level. What you're seeing are rate-limiting steps. Because if we go back and we think about the model, it's not necessarily that we're overfitting. It's that biochemical processes are actually, I guarantee you, it's way more than 3 processes that have to happen for the catastrophe to happen. Because there's, all sorts of configurational degrees of freedom in these proteins. These are very complex molecules. And so maybe a rotation of a beta sheet compared to a alpha helix somewhere in a protein has to happen. In other words, there's all these processes have to happen, but they happen at much faster timescales. And one of the things that we know from, again, century of study of chemistry is that some of the chemical processes, because there's a huge range of timescales of chemical processes, it's only the slowest ones that actually have an impact on the actually observed overall rate. And so this is a special kind of overfitting. It's an overfitting that actually fits with the real physical intuition, which is that what we have for sure m equals far greater than 3. But what m equals 3, is that it's 3 rate-limiting steps. It's 3 rate-limiting steps that are governing microtubule catastrophe. And then the rates are roughly, or I guess the characteristic times are roughly 2.5 to about 1.7 minutes. And there's all sorts of faster things that certainly are happening, but it doesn't provide any more generative capabilities for the actual experiment that we did. And so if we wanted to get into what those faster processes are, we're going to have to design another experiment. So yes, this is exactly what you would say. You would say that there are 3 rate-limiting steps on the times. Well, so on the time scale of the observations, yes, but by definition, rate-limiting means that if what you're trying to observe, which is catastrophe, there are really only 3 that are that occur at rates that would sort of impart that time scale to the catastrophe itself. There are all sorts of other processes like molecular vibrations and all kinds of stuff going on, but those really don't contribute appreciably to the, because they're so fast, they don't appreciate to the actual time scale of catastrophe. So I think this is a beautiful example of, by doing generative Bayesian modeling, you're actually learning about science. And I remember, and this is not the first approach I took to solving this problem. And I remember, I'm showing you the polished version, but I remember when I first saw this and the rate limiting step alarm bell went on, I'm like, that's what I'm looking at. Of course, I have 2 degrees in chemical engineering. I should have known this a priori, but this is one of the beauties I find with taking a Bayesian approach to inference, to having a generative modeling approach to inference, is that you are actually really directly thinking and learning about how nature works. And this is why I think that the Bayesian approach to the physical sciences, chemical sciences, biological sciences, geological sciences, astronomical sciences, it's so valuable.
Yeah.
Anyway, that's what I had to share with you. The notebooks have all the code and everything. You guys can work through the stand models on your own time. But thank you guys so much for listening to me. I talked forever, but I'm happy to have any discussion or questions. I can hang out. I'm not that hungry yet. It's getting close to lunchtime here, but I'm happy to hang out with you guys for a while.
Yeah, no, what I love also is that in the Bayesian framework, especially if you apply the Bayesian workflow, here we saw that it's not in a way, it's not an automated, statistics. Like, you didn't look at R hats or stuff like that. Like, often students ask me about, like, what number should I look at to see if my model is good, or not overfitting or things like that. And often I answer what I can only imagine is a very unsatisfying answer to them, is that usually you're more going to look at plots, and doing posterior predictive checks, for instance, is extremely important. And here we saw that you basically had the insight about this overfitting thanks to posterior predictive checks. But the cool thing is that if you apply the Bayesian workflow, it's like the Bayesian framework is demanding. Like, it's not gonna let you slack off and basically go on with a model that doesn't really work. If there is a really big problem, usually you're gonna end up seeing it, and then it's a choice of modeling whether you can live with this problem or not. But at least it's harder to put the dirt under the rug, I would say.
It is, and I think it has to feed back to your experiment as well, right? Like, if you're, it happens so often that you'll do an experiment, you'll do your inference, and then you'll be like, wow, my experiment just can't answer the question I sought to answer. I have to redesign my experiment, right? That's just
True.
Just happens. Okay, there's a question here that says, you did not model or try to estimate experimental noise. I usually include a noise term in my modeling. Any reason why you did not? The reason for that sort of it's twofold. So if we, I'm gonna go back. Oh, come on, this one. Go back here. Yeah, it really comes down to this, which is that when you're looking at the catastrophe event, it can't be any clearer, right? Like, it's like, boom, it's gone. And so whatever variability is present in that, we are actually explicitly modeling that there's variability by virtue of the fact that we're actually using a probability distribution, right? That there's variability by the nature of the fact that it's a Poisson process that's going to arrive at different times with no memory. And so that variability is already sort of baked into these types of observations. Now, there could be, I think by experimental noise, what you mean is like measurement issues. And yes, we could put that in there. In fact, one of the things that I considered in this was explicitly modeling one of the aspects of the measurement, which is that you can't actually observe the exact time at which catastrophe occurs. The reason for that is that you have a finite frame rate, right? You have to take a frame every few seconds, and I think their frame rate was something like once every 5 seconds, and you're actually really limited on that by experimental constraints. There's a phenomenon known as photo bleaching, which means that if you expose because these things they have what attached them, these molecules called fluorophores, which enable you to see the molecules. They're like basically little glowy molecules that are attached to the tubulin. And if you hit that with too much light, meaning if you keep taking images of it, they get destroyed and they don't glow anymore. And so then you just lose track of the microtubule. And so you can't actually image at a rate much faster than this because you're going to then lose your signal entirely. And so I even looked at, okay, well, we don't know. We only know that the catastrophe happened somewhere between the frame where the microtubule was there and then the frame where it was missing. And we don't know where in between it was. And so one of the things that I did with this data set was I did some analytical work where I actually looked at, okay, this is actually a discrete distribution because we're not actually observing the time of microtubules, or sorry, the time of catastrophe. We're observing the time at which we took a frame that we noticed that it was gone. And it could have happened anywhere between there. And then you can actually derive a probability distribution for that discrete distribution. And then you can compare what you would get inferentially by making the approximation that no, it's actually still continuous even though it's discrete. And the error in that is negligible. And I actually worked through that. I didn't include that in the notebooks. So yes, you always need to be cognizant of the measurement noise, but you should also model it generatively. And in some cases, and I won't get into it here, but there have certainly been cases in cellular biology stuff that we've looked at where the measurement noise is in some senses unmodelable and will destroy any model you're trying to do, which means you have to go back to the drawing board on your experiment. And so I think that modeling experimental noise is really part of this very important feedback between the modeling and the experimental design.
Nice. Thanks a lot, Justin. Thanks a lot for all these questions, folks. Any last questions before we let Justin have some lunch?
Okay.
Yes.
So I'm wondering why increasing m beyond 3 doesn't reduce the residuals. Is this the sign that there are additional physical processes in the system that cannot be explained by, I think you mean this multi-step distribution as opposed to the gamma. Right. So if we, I'll just quickly put that graphic back up so that it's in mind while we're talking about it. Oh, there it is. Okay. Okay. So, right. So we're not doing any better than 3 with 4, 5, or 6. Maybe just a hair with the 4, but nothing that we would ever, write home about. So why aren't we getting any better? And that really seems to be because if we look at what has to happen for these timescales, they're just, you have these 3, you're just getting shorter and shorter and shorter, and they don't impact the data at all, right? Because the timescale of the measurements, like we saw, is somewhere like the shortest one. If we go back to the EDA, the shortest one, here it is, was, by the way, I'm a big fan of bokeh. That's why I use that for all the plotting. The shortest observed microtubule catastrophe was just under 30 seconds, right? Or, half a minute. Now, if we go here and we look at the time scale of the next, of the last one, right? Most of the probability mass is below half a minute, right? There's just a hair of probability mass above half a minute. And that's probably why it's just ever so slightly better here. But beyond that, it can't have any impact on the data. So it's your residuals aren't going to go down, right? Because you can have all those things happening and they're just not going to affect the measurement. And again, that's straight up. I can't remember who the chem bio person was, but that is straight up the definition of a rate limiting step. And that's what you're just, a rate limiting step is staring at you in the data. I love it when your data speak. And that's one of the things that modeling can do, is they can help your data speak. I'm going to show you something really quickly here that it's my favorite data set of all time, and I had a former student and TA make this for me. So she sewed this. This is a data set of. She didn't label the axes because she knew that I would know exactly what it was. These are on the y-axis is the depth of beaks of Darwin's finches on the island of Daphne Major in the Galapagos, and the y-axis is the length of the beak. This particular data set I use with my students. We do a full analysis on it, where the data speak to you and they actually show you evolution happening in real time. Like you watch evolution happen in the data. And this, I love it because I love it when that happens, when the data tells you something about science. And that's what we saw here, is the data spoke to us. They told us, hey, there are rate limiting steps. And in this case, there are 3 of them. And that's what our analysis got us directly from the, we gave the data the microphone with which to speak. And it made me really happy. So this is like one of my favorite data sets of all time. Also, this microtubule catastrophe thing. It's so simple and rich.
Yeah,
and the frequentism makes it
cool.
hard as hell.
Yeah.
So
yeah. Yeah, thanks a lot, Justin. That was
My pleasure.
really fascinating. As you could saw in the prior elicitation part of the webinar, I didn't know anything about that. So I definitely learned a lot. I'm pretty sure that was the case for a lot of people here, even though we did seem to have very specialized biochem people in the chat. So that's really cool. Thanks a lot, people, for chiming in and making me look really bad. I always appreciate that from listeners. No, seriously, thanks a lot to all of you. Of course, big thank you to Justin. I'm going to work on putting that recording as soon as possible online, so that you folks who were in the webinar will get access to it as soon as it's ready. And if you're a patron of the show on the full posterity tier or higher, you'll get access to it right when it's out, or if you're in the webinar. Otherwise, you'll have to wait a month to get access to it. And I'm going to put the links about what we talked about today. So mainly the GitHub repo, where you have everything that Justin put together for us. And thanks, everybody.
There's also a quick question. There's a link
Matt
to the mitosis. Yeah, my link to the mitosis video. I'll make sure Alex has that, and he can post that for you. It's from a research paper. It may be paywalled. I hope not, but I'll definitely send the DOI for that.
Yeah, for sure. We'll put all of that in the show notes. Justin is gonna send me all he thinks is useful for those who want to dig deeper. Thanks again, everybody. And I think Virgil is here, and I think Virgil was one of the patrons who actually recommended me to have you, Justin, on the show again for a webinar like that. So thank you, Justin. Thank you very much, Virgil, for the recommendations. Actually, he sends me a lot of guest recommendations also often. So big thank you to you. Thanks for being so involved. And of course, if you folks have ideas of future webinars like that, let me know, because you see, I listen to you, so it might just happen. The next one will probably be in September, mid or end of September. We're going to have Benjamin Vincent again here, and we're going to walk through the do operator, the brand new do operator that we have in PyMC to do causal analysis. Yes. So to do causal analysis. But of course, get in touch with me for any recommendations that you folks have. Thanks again, everybody, for tuning in. I'm going to stop the recording now and wish you a very good day.
Let me show you how to be a good breezy and change your predictions.